So the next step is basically, we've identified a number of very important factors which regulate the gene, which define the gene regulatory network in specific types of leukemia, because they're different in each one of them. Right? AML is not just one cancer, it's many different ones. And each of them has a different phenotype and a different gene regulatory network, and it keeps itself alive by different means. So we have to identify all of these different factors which are specific for each type and what they're correlated with. And then we have to identify the genes which are downstream of those. And some of them are druggable. And we've shown in one case, when we've gone all the way from the identification of the mutation down to how the aberrant factor caused by this mutation functions, down to which target genes it interacts with, down to what downstream effects it does, down to an actual pharmacological target. So we can go all the way. And I think cancer research will go into this direction because we're going to have to identify what makes these different types of cancers tick. And that's not true just for AML, it's true for all cancers. We have to do this for all of them.